This program is made possible by the generosity of the New York Community Trust and the Pi Wackett Fund and one of our trustees, Wesley Janeway, so we're very grateful to them for their support. Following the program, there'll be a reception in the room where you were waiting, so we hope you'll stay around and join us. We have a terrific presentation for you, so I'm going to turn it over right away to our moderator, Dr. Max Gomez. Thanks for coming.
Thank you, Kathy, and good evening. How's everybody? Ready to learn a little something today? So it is appropriate, I think, that we are here tonight on, believe it or not, International Rare Disease Day. And yes, there is such a thing as International Rare Disease Day. By the way, it's also National Public Sleeping Day today, which, for real, but I hope that's not why we are here tonight. The main objective of Rare Disease Day is to raise awareness amongst the general public and decision makers about rare diseases and their impact on patients' lives. But you may ask, if these diseases are so rare, why are we focusing on them? Shouldn't we really be spending our time, energy, and dollars on the biggies, cancer, heart disease, Alzheimer's, AIDS, just to name a few? Well, tonight we're going to explain why research on rare diseases is indeed important and even essential. First, some numbers though. There are about 7,000 known rare diseases. A disease is considered rare in the US when it affects fewer than 200,000 people, which doesn't sound all that rare to me, but that's the American definition. Worldwide though, rare diseases affect about 300 million people. 1 in 20 people at some point in their lives will live with a rare disease. 50% of rare disease patients are children. Now, all that indeed does make it sound like the impact is widespread. But you may also ask, why are we talking about these diseases at a genome center of all places? Well, that's because 80% of rare diseases have identified genetic origins. The others are the result of infections, allergies, environmental causes, and so on. The good news is that there are new advances in understanding the genetics behind rare diseases, especially the pediatric ones. Tonight we'll hear how next-generation sequencing, and yes, we will define that for you non-molecular biologists, how that is uncovering the genetic variants responsible for rare disease risks. Also, we'll talk about how genetics may actually be relevant to understanding infectious diseases and heart defects in children. And most importantly, we'll answer the first question I posed about why we should all care about rare diseases, how the genomic study of rare diseases has furthered our understanding of all diseases. And that's why we're here and why it is important. So without further ado, since we have a lot to cover tonight, the format, just so you know, so you're not surprised, I will introduce each of our panelists. They'll make a brief presentation. We'll ask a question or two, and then at the end, we'll bring them all up and open it up for questions to our audience. Now, as you're thinking about questions, I've done this a lot before, and I always get scared when I hand microphones to people and turn them loose. This is not a political rally. You're not here to make a speech, so please actually have a question, and then we'll figure out who our best panelist is to answer that. But actually, I think all of our panelists will probably chime in. So let me begin with Dr. Tuuli Lappalainen, who's a core faculty member at the New York Genome Center. She also holds a joint appointment as assistant professor in the Department of Systems Biology at Columbia University. Her research focuses on the study of genetic variations in human populations and its contribution to traits and diseases. And this evening, Dr. Lappalainen will discuss how next-generation sequencing, and you get to define that, how next-generation sequencing is uncovering the genetic variants responsible for rare disease risks. Dr. Lappalainen.
Thank you, Max, for the introduction, and thanks for coming here. And as you see, my title is not exactly how Max described it, like the title that I gave to Max initially. I changed the next generation sequencing to novel sequencing approaches. And the reason for that is that we have actually been doing next generation sequencing for about 10 years now. So it's not really next generation, it's this generation sequencing. And that's one thing that makes it so exciting. And basically what I want to talk to you very briefly now in a couple of slides is how these sequencing approaches have enabled genetic research of rare diseases, and very briefly about the things that we have learned and the next challenges in the field. So it's widely known that genetic variants, the tiny genetic differences between us, contribute to human traits and diseases. We all know that children tend to be a little bit like their parents. And in the field of human genetics during the past 10 or so years, we've been very successful in mapping these specific genetic variants that contribute to different traits and diseases. And kind of like on the opposite end of the spectrum of human traits compared to rare diseases are traits like height, where we have now identified about 700 genetic variants spread across the genome, across all the chromosomes, where each of these variants has a teeny tiny effect on height, and the total the height that you end up having is a composite effect of those variants plus environmental effects, like let's say your nutrition as a child. But then the other end of the spectrum are really rare diseases that are often quite sort of severe phenotypes, and that are often driven by mutations or variants in one or very few genes. So this is, I just took one example here, fibrodysplasia ossificans progressiva, which is a quite devastating disease that leads to sort of bone formation in soft tissues, which is caused by mutations in one specific gene. And so and of course there are a lot of diseases and traits that kind of fall between the spectrum of these two kind of extreme examples. And as Max said, rare diseases are diseases that affect less than 1 in 2,000 individuals, but of course, altogether, even though each individual disease is rare, altogether they affect tens of millions of people in the US. And these are often genetic, so these diseases can be caused by genetic variants that are inherited from the parents, and sometimes genetic variants that are new mutations that parents didn't carry. All of us carry some mutations that are basically new mutations that happen between our parents' generation and us. And then if you happen to be unlucky, those mutations sometimes might actually break a gene in a very bad way that leads to a rare disease. So to talk about how the novel sequencing approaches have advanced the study and diagnosis of rare diseases, as a little historical timeline. So in the sort of since late 1980s or so, geneticists have been able to have this kind of sparse maps across the genome and then do very targeted analysis of a few genes to discover and also diagnose rare diseases. For the past 10 or 8 years, since the development of what then was next generation sequencing, now high throughput sequencing, we have been able to do sequencing analysis of all the genes in the human genome, which of course gives us a much denser map of data to find the contributions of genetic variants underlying these rare diseases and to diagnose them. And for the past couple of years, there has been tremendous success in our ability to sequence not just the genes, but actually the entire human genome that enables even deeper analysis. And this has led to huge advances in the research and diagnosis of rare diseases. So Every year, there's been a discovery of novel disease genes for a couple of hundred different diseases, or forming those links between gene disruptions and diseases. And this has been really important for understanding the biology of human diseases, especially in rare diseases. There is, if you are able to identify what is the gene that has broken, that has given rise to this disease, it gives you an instant window into what might be going wrong in the cells, and that affects the anatomy and physiology in a way that gives rise to this disease. It's also extremely important for human genetic research, understanding how our genome, how our genes actually work to give rise to a functional individual. In humans, we cannot really sort of do experiments like we might do in mice or in worms, where we would just kind of like manipulate the genome and see what happens. But when we survey the naturally occurring genetic variation in human populations and individuals with disease, we can actually develop an understanding of how little genetic perturbations affect human diseases and traits. And of course, understanding this deep biology of human diseases also gives us the tools and the insights that is necessary for development of novel treatments. And I won't really talk about the clinical side of things. I think Bruce and maybe Jean-Laurent will talk a little bit about that, but having these sequencing tools has also enabled a much more efficient diagnosis of rare diseases and discovery of those genetic variants that underlie an individual patient's condition. So as a genetic researcher, we're always thinking about kind of not only the advances that we have made, but the next challenges, and then how we can kind of like push those boundaries of enabling even better understanding of rare diseases and well, all kinds of diseases, as well as the conditions of individual patients. And here I want to kind of just give a brief overview of some of the challenges that we still have in the field that all of us are working really hard to solve. So one of the kind of situations that you might have if you have a patient and you sequence the genome, you find a mutation in a known disease gene, but you're not necessarily sure if it actually disrupts the gene, if that is the mutation that is actually causing the disease. This would be called variants of unknown significance, and it's a big challenge for all of us. And there is a lot of work in laboratory experimental kind of methods development and statistical methods development to be better able to characterize and distinguish the disease-causing mutations from the ones that don't actually have a big effect. The same applies for mutations that are outside the gene that might not necessarily kind of disrupt the gene itself, but may potentially kind of turn it off, affect its regulation. And here there is a lot of work in measuring not just the genome in the patients, but also gene expression and other molecular phenotypes. And this is something that my laboratory works on as well. There are also situations where the known previously discovered disease genes don't actually carry a mutation in a given patient. And then there is the question of like, is there an undiscovered disease gene, a gene whose disruption actually causes disease, but we haven't kind of made that link yet. And there are tremendously big, successful international collaborations for building larger studies to be able to discover and map these disease genes for individual diseases. For rare diseases, it's particularly challenging because the number of patients is small, and this requires large collaborations between different laboratories and also internationally. There are also situations where the patient actually has a mutation, but our analysis hasn't been able to find that mutation. Maybe our sequencing data wasn't good enough, maybe our analytical methods kind of failed there. And this is, of course, one area where we do a lot of methods development to be able to find all these mutations. We have a lot of disease situations where different individuals may carry even the exactly same disease-causing variant, but may have different disease outcomes, different severity of these diseases. And there is one of the areas that my laboratory works on a lot is understanding these genetic and environmental modifiers that contribute to these things. And finally, if the disease is not even genetic, it's also really important to understand environmental factors underlying rare diseases. And finally, what I want to finish with is just a pointing back to the fact that ultimately creating this genetic understanding, genetic maps of rare disease is really important and gives a platform that we can use to develop interventions, medications, and other forms to actually help individuals with rare disease. I will end here and take questions from Max right now.
That's right. So Tuuli, let me make sure that people, I think, understand here, because what you're saying is that when you do this sort of sequencing, you'll find there are thousands and thousands of variations in our genomes that are completely normal. They're just different. You've got blonde hair, blue eyes. Clearly, I don't. And those are going to be normal variations. So how do you sort out these normal variations and from disease-causing variations or actual mutations?
Yeah, it's a substantial challenge, but we have a couple of things in our toolkit how we can do this. Specifically, in the case of rare disease, first of all, you would need to collect data from a relatively large number of patients, basically as many as you can get, and then compare the genomes, the genetic variants found in those patients with large numbers, nowadays even tens of thousands of genomes from normal individuals, to be able to find those kinds of variants that are extremely rare or even completely absent in healthy individuals, but then present in patients. And there are different kind of sort of filtering algorithms that we use to narrow down to kind of genes that are particularly sort of like vulnerable to mutations, and mutations that based on kind of like our prediction of how they break the gene may have an effect.
Because you have an idea already of some genes that are probably more, they're higher suspicion that they may be involved in the disease, but it's also why it's important to gather as many as you can of people with these particular rare diseases so you can compare them, right? Yeah,
exactly. It's having basically as many patients as you can. The more the better. That is really one of the basically key factors in genetic research in determining how many discoveries you are able to make, to have a large number of patients, and then also to have a large number of healthy individuals, because in many ways our understanding of what is disease or what does a kind of a disease-causing variant look like is based on our understanding of what do the genomes of healthy people look like. We need to be able to make that comparison.
And that's all been enabled by this high throughput gene sequencing, because it used to be a big deal to have to sequence even one genome, right?
Yeah.
Now you can do it much more easily, and now you're more able to compare them.
Yeah, exactly. And in addition to being able to sequence those genomes, you also need to be able to bring those genomes together in one place. And that is kind of something that many people in our field work on, basically building these large collaborations, collaborating with large number of different laboratories to kind of bring these data together to be able to analyze it jointly.
Terrific. Well, thank you. I'm sure the audience will have many more questions relating to your work there, and we'll get to those in a second. Make notes of them, by the way, so you don't forget them, guys. Let me introduce our next panelist, is Dr. Jean-Laurent Casanova. Is that a great name or what? Is a professor at the St. Giles Laboratory of Human Genetics of Infectious Diseases at the Rockefeller University, one of my old alma maters, and an investigator at the Howard Hughes Medical Institute. His research focuses on human susceptibility to pediatric infectious diseases. This evening, Dr. Casanova will discuss genetic theories of childhood infectious diseases, unless you change your title like Tuuli did for me.
So what I will discuss with you is simple. I will discuss the idea that infectious diseases that you think are infectious, caused by microbes, in fact are also genetic disorders. And second, the idea that rare infections are rare diseases, that is obvious, but that common infections, because they can be caused by genetic disorders, in fact, can be seen as rare disorders, rare diseases, because a common infection, if it is caused by a myriad of rare disorders can be seen as a collection of rare diseases. So I will, before discussing with you some of these infections, rare and common, and their genetic basis, I will give you a background so that you understand how investigators came up with the idea that infectious diseases could also be genetic disorders. And we have to start with the beginning, and the beginning is 1866, when the two fundamental theories that have structured biology and medicine were documented and published, the germ theory by Pasteur and the genetic theory by Mendel. From 1866 onward, all physiological and pathological variations across living organisms, including in humans, were ascribed to germline genetic variations or to the impact of the environment, including in particular the impact of invading microorganisms. So these two theories were seen as mutually exclusive until the turn of the 20th century, when it was discovered that most humans are infected by most microbes and do okay, and that for any given pathogen, only a small minority of infected individuals would develop life-threatening, which in these years meant lethal, infectious disease. And the reason why half of the children died before the age of 15 years, resulting in a life expectancy at birth around 20 years, and it had been so for 200,000 years worldwide. The reason underlying this massive burden caused by infectious agents was not that the infectious agents are intrinsically pathogenic, but it is that there are hundreds and hundreds of them. And if each of them kills 1 in 1,000 or 1 in 10,000 people, you can lose half of the population before the age of 15 years. So the problem that was posed 100 years ago is what are the other factors that cause a particular virus or bacterium or fungus or parasite to kill 1 in 100, 1 in 1,000, or 1 in 10,000 infected individuals. And the solution was quickly apparent to geneticists, human geneticists, both population geneticists and pediatricians, classical Mendelian clinical geneticists, and that's known as the genetic theory of infectious diseases. These investigators proposed that a child, an adolescent, or a young adult who would die of infection would die not only because of the infection, but also because of a genetic vulnerability that makes this particular individual prone to severe infectious disease. So the idea was proposed, and it was documented clinically and epidemiologically from the 20s to the 50s. And then between the 50s and the 90s of the past century, rare cases of primary immunodeficiencies, that is, rare diseases underlying a variety of infections in affected children, were documented. But it's only over the last 25 years that infectious diseases striking otherwise healthy children or adolescents were shown to be genetic. And there are two groups, and we'll briefly review these two groups to illustrate this general idea. The first group are the Mendelian infections. Mendelian means that the genetic disorder is caused by a single genetic lesion, and that all individuals carrying that genetic lesion have the disease. So we refer that as complete penetrance. And there are a number of Mendelian infections that have been explained at the molecular and cellular level over the last 20 years. Mycobacterial diseases, in particular those caused by poorly virulent mycobacterial species that you can drink in tap water, or the BCG vaccine, were shown to be caused by inborn errors of an immunological circuit controlled by a molecule which is gamma interferon. Lethal disease caused by the Epstein-Barr virus was also shown to be caused by mutations in a gene called SAP and other genes that are related to SAP and that permit the control of this EB virus in the blood circulating B cells. A third disease that is a Mendelian infection is epidermodysplasia verruciformis. These are children, adolescents, or adults who are covered with flatwarts that transform into non-melanoma skin cancer, typically during the second or third decade of life. And this terrible disease, it's a rare disease, a very rare infection, was shown to be caused by mutations in genes that control how keratinocytes, the cells that make the skin, how these keratinocytes can control the papillomaviruses causing lesions in these patients. A fourth Mendelian infection is chronic mucocutaneous candidiasis. These are patients who develop fungal lesions of the nails, the skin, and the mouth, sometimes the esophagus. And these patients were shown to carry mutations in another immunological circuit controlled by interleukin-17. And the other fungal disease that was shown to be genetic is invasive dermatophytic disease. So dermatophytes like Candida are typically commensals. We typically carry them on our mucosae or on our tegument, and rarely they can cause disease, sometimes aggressive disease. This is rare, and it is caused by mutations in another gene called CAR9. And the sixth and last Mendelian infection was more recently described, and these are patients who develop encephalitis, that is an infection of the brain, caused by common viruses. It can be the influenza virus causing flu, or it can be herpes simplex or other viruses. And this is caused by mutations in a circuit that controls the cell intrinsic immunity against viruses in that particular region of the brain. But Mendelian infections are rare. You know, if infectious diseases segregated like the color of the eye or the color of the skin or the quality of your hair, well, you know, that would be known. Most infections do not segregate like genetic disorders. They don't. They're typically sporadic. They strike, you know, a child, an adolescent, or an adult. These are typical cases. Someone with severe flu or severe herpes encephalitis or severe tuberculosis. And the idea that has emerged over the last 20 years is that these infections are not Mendelian, but they're monogenic. That is, they can be caused by single gene lesions, but most individuals with these single gene lesions, for reasons we're not going to discuss today, most of these genetically affected individuals do not present with the severe infectious disease considered. And I will quickly mention a few examples. Herpes simplex virus encephalitis strikes about 1 in 10,000 children. Most children are infected by herpes simplex. Typically it causes cold sores, but rarely the virus goes to the brain and causes herpes encephalitis, which is lethal, or for most survivors, sadly leading to severe, profound, chronic neurological sequelae. And this condition was shown to be caused by mutations in another circuit that controls immunity against viruses in the brain. Another infection, invasive pneumococcal disease, was shown to be caused by mutations that control the invasion of these bacteria through mucosa. Staphylococcal disease, another infection you're familiar with, that can cause, for example, furunculosis or folliculitis. Well, the severe forms of staphylococcal disease have been shown in some children to be caused by mutations in a related circuit that controls how the natural barriers, together with leukocytes, prevent the invasion of the organism by these bacteria. Flu is a common infection with which you're familiar. Most of you have had flu. Very rarely, flu takes the patient to the intensive care unit, and that is unexplained, or it was unexplained until the first cases of genetic flu were discovered. It was shown that, you know, it was not a matter of bad luck, but it was a matter of being genetically predisposed to life-threatening flu. And a rare infection, which is also a cancer, Kaposi sarcoma, caused by another herpes virus 8, which is exceedingly rare, especially here in the US, was shown in a small proportion of cases to be caused by other inborn errors of immunity. And I will close with an infectious disease with which you're much more familiar than Kaposi sarcoma, which is tuberculosis. And it has gradually appeared that this common infectious disease that has killed millions and millions of humans, and which is still today the most dreadful infectious disease worldwide, that this condition, in at least some cases, can be caused by genetic lesions that are individually rare. And you have here on the slide 2 autosomal recessive disorders, each found in 1 in 100,000 people, but each of which is a cause of TB, provided, of course, that the genetically affected individual is exposed to Mycobacterium tuberculosis. So I hope I've convinced you that infectious diseases can be genetic, and that not only rare but also common infectious diseases can be caused by a collection of rare genetic disorders. Thank you for your attention.
So Jean-Laurent, let me make sure that I understand this. So we have the Mendelian and the sporadic versions. Are many of these what you would consider normal genetic variants then?
So they're rare genetic variations. They occur in 1% or 0.1% or 0.01% of the general population, and their combination is even more rare.
And then if you have one or more of these variations that you're talking about, you have a predisposed susceptibility to a particular
infection. For example, if you're mutated in a gene which is RF7, you're completely fine until the moment you're exposed to the flu virus.
So it depends on what infectious agent you happen to bump into.
Exactly.
Would any of these variants then explain or potentially explain the different immune responses to vaccine, for example?
So this is an important question, which is related, as you point out, but that has not been studied. So the genetic control of responsiveness or lack of responsiveness to vaccines has not been deciphered in molecular or cellular terms.
But that's at least a possibility.
Like everything, it is probably genetic.
Like everything. Thank you, Jean-Laurent. And last but not least, as we say, we have Dr. Bruce Gelb, who is the Global Family Chair and Director of the Mindich Child Health and Development Institute and the Professor for Pediatrics and Genetics and Genomic Sciences at the Icahn School of Medicine at Mount Sinai. You get that all on a card? Okay. His research focuses on using genetic approaches to understand the causes of pediatric cardiac defects and hereditary heart diseases. And this evening, Dr. Gelb will discuss the latest advances in pediatric genomic medicine. Dr. Gelb.
Okay, good evening. Happy holiday to all of us who celebrate it. I appreciate the opportunity to be here and to the organizers for putting it together. As you'll hear, I've been working on a couple of projects actually with the people here at the center, so it's great to be down here. So I am going to talk about precision medicine, and particularly as it applies to children, so really pediatric genomic medicine, and that's because for the most part, in that instance, we are talking about the rare diseases that we're celebrating here today. So first I'll start with precision medicine. I'm not going to take a poll, but I bet if I did, that everyone in this room has heard of precision medicine. And I don't know how many people we are, but over 100. If I asked everybody to define it, whether you have a science background or not, we'd probably come up with close to 100 different definitions. And that's the problem we're having. There isn't one, but I'll just give you one, both from Francis Collins and Harold Varmus. So that's reasonable. And that is that precision medicine is preventive and treatment strategies that take individual variability into account. Now, as we think of precision medicine, what is widely conceded, and I believe to be true also, is that cancer is the front line on this. We are clearly learning that different cancers result from, or the same cancers can result from different mutations. The responses to chemotherapy varies, and so on and so forth. So where do we go from there? And my answer is, if we concede that cancer is the low-hanging fruit for precision medicine, the second lowest hanging fruit, if you will, is pediatric medicine. And that's for the reasons that Max referred to earlier. A lot of the disorders we know about that occur in children are genetically driven, and even those that we didn't necessarily think of as genetic, as you just heard from Jean-Laurent, actually are turning out to be genetic. And in fact, it's because of the fact that the genetic factors are quite strong that lead to those disorders. That's why they're presenting in childhood. It makes them both easier to identify and easier then to think about therapies based on the genetic information. It gets murkier. Obviously, the numbers are bigger when you think about some things like Alzheimer's, but it gets murkier once you move into adulthood. So you've already heard these numbers a bunch of times. I'm a little more, depending on your perspective, optimistic. I think there are going to be more Mendelian disorders than the 7,000 quoted. And it's exactly for the reason that Jean-Laurent just educated us about. We've gone from, when I was a medical student, knowing of about 3 or 4 immunodeficiencies to now something over 300. And I think we're going to see a lot of other things expand. And so I think there's a lot more of these. Now, you've already seen this figure in Tuuli's talk, and we obviously didn't coordinate in that sense, but that's okay, because I want to take a moment to just point out a couple things about this. This is a 30-year timeline, and what's being shown in the bars is how many of these rare disorders were figured out in any given year. And this is actually when I started getting into genetics, and we were really slogging along there trying to find these using what was then called positional cloning or reverse genetics. And what happens in here is, as the human genome gets sequenced, the whole thing accelerates massively. And that's what's going on in this period. And you can see that we got up to something around 200 a year. And as Tuuli mentioned, we've now jumped up and we're approaching something closer to 300 disorders solved a year. And that's because of the next generation sequencing that has really made that possible. So that's great on the research side, and I focus a lot of my efforts there. But what I really want to talk about is how I think that's going to change medicine, at least pediatric medicine. So currently, the way medicine is practiced in general in pediatrics, including pediatric medical genetics, is shown here in the top. We see a child in whom we suspect strongly that they have a genetic disorder. We do some sort of genetic testing. Some of you will know the details of that are going to look at some sort of problems. If we know some of the genes for the trait that we're interested in, we can then go on to do some sort of resequencing of some number of genes. Could be 10 genes, could be 100 genes, depends how many we know. And then sometimes, and we don't always make it through this last arrow, if we're stuck, we'll go on and then sequence the whole collection of genes, which is, as Tuuli correctly described for you, whole exome sequencing. And now we're actually even starting to do whole genome sequencing. And that's simply because the price has come down so much to enable that to be possible. But that's not going to be the way it's going to move forward. What is starting to, and by the way, this approach results in solving something like a quarter, maybe a little bit, a third of the cases. And the problem is that our patients, in the meanwhile, are on a diagnostic odyssey that can last certainly for months and often for years. So the new model that groups are starting to explore, and I think is coming increasingly possible, looks like this. We see the child, we suspect the genetic disorder, and rather than doing a whole series of tests, we immediately go to the test that we know that can most likely get us to a solution, and that's going to be exome or genome sequencing. And so that means, instead of it taking months to years, that this can take weeks in many instances, and we can solve maybe up to a half of the cases. And I think over time, as we learn more and more about the genome, we will solve higher numbers. So let me give you an example. We actually run a program at Mount Sinai where we deal with this sort of thing, an undiagnosed disease program. And let me share with you a case of one of the first patients we saw last year. It was and I want you to think as I'm going through this and tell you a little bit about all the problems she was having, just imagine how many physicians must have been involved in her care and other care providers too. So she was an 18-year-old girl who had really presented in infancy with problems, and she had a fairly complex picture that included a kind of kidney disease where the body was attacking her own kidneys, ultimately destroyed those kidneys, resulting in the need for a kidney transplant. She successfully underwent that transplant, and unfortunately, the exact same disease recurred, destroying the transplant kidney. That's terrible. So when we first met her, the kidney had been more or less destroyed, and she was back on dialysis, which, if you've had any personal experience with, you know is a very tough way to go. On top of that, this young lady has a chronic problem with inflammation of her gastrointestinal tract, specifically of the large intestine. She's had that for years. It's not the typical type that people in New York might know about, like Crohn's or ulcerative colitis, a nonspecific inflammation. And she had a skin rash that kind of comes and goes. It had been biopsied. Nobody could quite figure it out. And then when I met her, I noticed, and this was a tip-off to figuring out this case, that one of her legs was a little bit bigger than the other one. So again, she's 18 years that her family has been coping with this. So we went ahead and did exome sequencing, and in well under a month, we had figured out that the underlying problem in this young lady was a mutation in this gene. The specifics aren't important. The important point is that this is a gene well known to cause cancer, be involved in cancer. And in fact, the mutation that she had been born with that had come, since before birth is one that's been tested, seen in cancers, and already tested for the protein. So we knew that this was a hyperactive protein. So we went to the literature and said, well, has anybody else reported an individual who's born with mutation in this gene? The answer was no. And that was until last summer, when a Canadian family was reported 2 generations, a mom and 2 of her children, overlapping kinds of problems with the GI tract and so on. And although not the exact same mutation, the same flavor of mutation in the exact same gene. And the beautiful thing about this, and this is why this is very exciting kind of work, is that for cancer, the drug companies have already developed something and has been FDA approved that can be used for tumors where you have these kinds of mutations in this gene. And so you'd say, well, could this work in the patients? I don't know yet. We're just starting our patient on such a drug, but I can tell you the Canadian family was started on one of these drugs. There are 2 of them on the market with remarkable resolution of their medical problems. So that's what we can do with this genetics first type of approach to medicine. And so right now, in a project, we're trying to bring this to New York. So NIH, and specifically the National Human Genome Research Institute, NHGRI, has a program called CSER, where they're trying to test how can we bring the kinds of exciting genetic sequencing I just talked to into day-to-day medicine. It's not trivial. Most doctors don't know how to use this information, et cetera, et cetera. And so we have started a project that is a collaboration between 3 places, Mount Sinai, where I am, Montefiore, and here at the New York Genome Center, and that's why those logos are up. And the name of our project is NYC KidSeq. And so what we're just starting to do is to test how does genome sequencing work in the real world if you don't do it in the research, if you do it in a real clinical arena? And most importantly, how do we explain to people, some of them having college education, some of them not much education at all, how good a job can we do? How do we properly explain to people all the things we're finding? Because when you start doing this, you find stuff that's of interest for the disease, but you also find other stuff that people may or may not want to know about. For instance, a tendency to get cancers or something like that. And so what's listed here are the things we're starting to do. And our ultimate goal and what we're going to do is recruit over 1,000 kids, over 1,100 kids actually, with problems in 3 areas, either neurologic problems like epilepsy, seizure problems, immunologic problems like the ones you just heard about from Jean-Laurent, or the ones that are the most important because I'm a cardiologist, which are cardiac problems. And so we're going to do that in 2 places, and we're going to see how useful that winds up in a clinical setting. So this is important, and there are other sites in the country that are testing this for other diseases, some of them in kids, some in adults, some of them in cancer, others germline genetics as we are. And I think that's going to move us forward to figure out how do we deliver the information to everyday people who have no medical training? How do we get the everyday doctor to understand these kind of testing to really transform medicine? So I'm going to stop there. Thank you for your attention.
Let me ask a couple of questions. One that is sort of just simple and explanatory, I hope. You use the term exome where most people have been using the term genome.
Sure.
What's the difference?
So the genome refers to, people may know that if you take the full collection of our chromosomes, each is made up of a series of DNA bases, and in total, they are 3 billion bases. So that collection of everything, the whole thing in our species is called, that's the genome. It's 3 billion bases in our species. But a lot of the sequences are either controller sequences, Tuuli spoke about that, or so-called junk DNA that we don't even know if it does anything, although we were suspicious over time we'll realize more and more does. But the percentage that actually has the instructions, the instruction manual to make all the genes, to make all the proteins that our body ever makes from earliest development to the end of our lives, that only makes up about 1 to 2%. Because those genes are broken up into little blocks that are referred to as exons, the collection of all the exons for all the genes is referred to as the exome. And it's about 1 to 2% of the genome. So that the point is that if you can sequence just that, you get a long way towards what you can actually use in a clinical setting right now, because we're still working hard to figure out what changes outside of the coding instructions mean from a disease point of view or just even normal function point of view.
Now, I have a lot of questions myself, but if there's somebody there while you're getting the microphone to them, let me ask Tuuli the one quick question, because I want to make sure that people also understand what is the value of finding, let's say, a single gene that causes a disease if we can't go in and repair that gene.
Yeah, so there is a sort of general value for genetic research of sort of like understanding what does this gene actually kind of do in your body? What does it do in our cells? Which is sort of one of the fundamental building blocks of like biology and understanding how does our body work? How does our genome work? Then from a more practical perspective, and Bruce mentioned this, there is the kind of if you have identified a gene that where mutations, lesions in the gene cause a particular disease, that enables you to use that information to better diagnose patients, and that can be tremendously important even in those cases where there is no targeted drug available. It ends this diagnostic odyssey. It gives the families and the patients information of like, okay, this is what has been wrong all these years, and now I know, and that's often quite important, even if targeted treatment is not available.
And also figuring out what's the gene product, what does that gene make or doesn't make when it's a mutation. That helps us understand how it causes a particular disease.
Yeah, exactly. And then kind of like down the line, if there is no existing drug, maybe there can be. You can then, research community and drug companies can start working on developing those treatments.
Good. Let's go ahead and take a question over here. Yes, you have a microphone. Go ahead. Okay, stand up for me so we can see you.
You emphasize a lot about the variance of uncertain significance, but my question here is, with disorders such as microdeletions and microduplications, that the phenotype is not quite characterized, so there's some difficulty in the community as to whether or not to characterize them as a disease, such as 16p13.11. Where do you see this going in the future?
We have a ringer in the audience, apparently.
So, sure, I'd be happy to first maybe translate the question so everybody's with us and then try and answer it. So one of the things that has been remarkable that we've learned since the sequencing of the human genome, we always work from the assumption that, except for the sex chromosomes, that we have 2 copies of every gene. Everybody knows that, one from mom, one from dad. And so that's that. All the chromosomes would be complete. And then there'd be these rare individuals where a chunk of DNA would be missing, or perhaps an extra chunk, and that would lead to disease. But the fascinating discovery that was made was that that's not true. That in fact, somewhere between 10 to 15% of our genome is in play. If I tested everybody here today, we would surely find many places in all of our genomes where some of us have whole and some of us have extra copies. And some of these lacking pieces, for instance, can be pretty big, like 1 million base pairs with genes in it. And so that the fact that we're still seemingly having no consequences of that is rather amazing. So again, we've had to go through that problem of sorting out when we do have a child, and it started really with kids with neurodevelopmental challenges. It turns out about 10% of them are due to these sorts of problems. So the first thing is, once you test many people, the way Tuuli referred to, you get an idea of what's seen out there in the general population. So then when you see something that's really enriched for a particular group of children with a disease such as those with neurodevelopmental, and we have the same thing with children with heart defects, that gives us a clear sense that that is actually causing or contributing to the problem. And I'll just say one more thing about it is, where we've struggled, and the gentleman mentioned one particular deletion on chromosome 15, it turns out that when you look at kids with neurodevelopmental problems, we see it in congenital heart disease as well, that particular one is not, it's not that the parents didn't have it and the kid does. Often it's inherited. And so you're faced with a child who's got these challenges, whichever organ it is, and the parents are fine. But that's turning out to be not exactly right. So people have started, they did a beautiful study in Iceland, for instance, where they took a group of patients with specifically that change, and they tested they did cognitive testing on them. And while nearly all of them are in the normal range, as a group, they're shifted towards the range with lesser ability. So it's not completely without its effect. So we still have a ways to go to figure all that out. But as with variants of unknown significance, as we're getting more and more data on now hundreds of thousands of people, our ability to say what's really, really, really rare or never seen before is very helpful towards taking the step towards saying it's really causing the problem.
You answered one of my questions. Jean-Laurent, let me ask you, these genetic predispositions, if you will, to infectious diseases, the ones that have been identified, are they all part of what we would consider the immune system?
Well, in a broad sense, they are. Can you hear me? Yeah. In a broad sense, they're part of immunity, not necessarily leukocytes, but all cells of the organism contribute to fending off microorganisms.
And is it, if an individual has this kind of variation, is it heritable? Is it something then that you would presume the children would have?
Well, one of the two, as Bruce says, one of the two copies will be transmitted to a child, but then depending on who the partner is and whether the partner carries similar mutations, the children may or may not have the same condition.
And one or two copies may determine how severely impacted they are by
whether they have the genetic disorder or not.
Someone else, yes.
Hi. I just wanted to continue your question, actually, to Dr. Casanova. I just also wanted to mention there is a conference next week at Columbia that touches on infectious triggered and autoimmune diseases of the brain. So it's really timely that we're talking about this. But my question is, are you seeing this as it relates to strep? And it seems to me, just for clarity, are you saying that essentially you have potentially a genetic predisposition, so whether you're talking about flu or strep or any of these different infectious triggers, that it's the bullet, this environmental trigger, the bullet, flu, whatever, that then causes only in this genetically predisposed kid these symptoms to happen. So if that is the case, and I'd love for you to elaborate, is it a far cry, or is it, I mean, it doesn't sound like it's beyond the realm of possibility that strep could be one of those. And I hate to get into the whole, as you mentioned, vaccinations, and I'm not going to get into autism either, but I do want to ask if we know that it's part of, I remember an NIMH former director was saying, we just don't know enough information. Some people would say, well, my kid didn't get it and they had the vaccination, so therefore it can't be the vaccinations causing it. But isn't it possible that the genetic predisposition with the infectious trigger is why some kids have whatever and some don't?
Right. So there are several questions in your question. Let me try to take them one after another concisely. So I didn't say that all severe infectious diseases were genetic. I say that for all infectious diseases studied, genetic cases were documented. And we don't know the proportion of genetic cases for most infectious diseases. Okay? So second question is the strep vaccine. As I responded to our chairman and moderator earlier, the genetic basis of the response to vaccines is unknown. So the relationship between the genetic control of responses to vaccines and the genetic control of disease caused by the corresponding microorganism is unknown.
And let me just, to elaborate a little bit, because recently we've heard of some infectious diseases that have sequelae way beyond the infection. ADHD, other neurologic, behavioral sequelae from what seems to be something simple, well, relatively simple as rheumatic fever.
Right. So this is another important question. Could it be that the long-term consequences of infectious diseases are genetically controlled? It's unknown.
Interesting. Another question. We have one? Yes.
So, wonderful talks, everyone. I want to touch again on the whole genome and whole exome application in the large population, like the study that you're proposing in the kids. How do you see the complexity of interpretation of the data, which, I mean, we're touching on VUS, on next generation panels or novel sequencing panels, and in the genome and in the exome will be even more complex. And how do you think that the link with the clinical manifestations, especially for cardiac conditions that may be not as obvious as the pediatric whole symptoms of neurological malformation will do? And as a side also, do you see a role for transcriptome studies integrating the genetics? I know some companies are trying to do, at least for wherever the genes are expressed in peripheral blood or in organs that are easily biopsiable.
Yeah. So many questions, interesting ones. So it's going to be a while till we are able to fully analyze genomes routinely on everybody. But what we can do today is understand, when we look at coding variation, to a large extent, find genes. And the issue here is that it's getting cheaper and cheaper to do things like genome sequencing. And so at a certain point, it will stop making sense to just test for a subset of genes and go through all the process that a lab has to go through in order to do that, versus just sequencing the genome. And then as we see the problem, we look up the part of the genes that we're interested in. That's sort of where we're headed. I was referring to patients where they have things that we just aren't so certain about, and it's just a lot better to jump in early rather than doing lots of testing. And I only referred to, of course, genetic testing, but meanwhile, these kids are getting poked and prodded in lots of different ways, spending many days in the hospital, and all that entails. I think that's a lot more efficient. Where I think as a society we have to be very careful, though, is I am certainly not a proponent. There are people who believe in this, in just saying, okay, we'll just start doing a genome on every baby born, and then we'll wait and see what happens, and then look it up. I think we are quickly going to learn that what we understand about genetics, by virtue of having for decades studied people who have medical problems of one type or another and figured out their genetics, is not going to be quite the same for many, many diseases as just pulling out the genetics and then waiting for the person to have that disease. It'll work well for some diseases, like, for instance, sickle cell disease. If you go and just sequence that mutation and everybody and say, oh, that person has 2 copies, they will have sickle cell disease. But there are many diseases where that's not the case. The last thing you asked was about the transcriptome. So for everybody's benefit, what that refers to is doing sequencing, but this time, instead of sequencing the DNA, you're sequencing the RNA that are being produced by a given cell type. And there are increasing amount of scientific evidence that when you're looking for certain types of rare disorders, that looking for mistakes made in how the gene gets expressed, which is what you're measuring when you're measuring RNA, can give you a very big clue to where to look in the genome for the problem. So certainly people are showing that. I don't think it's that helpful, though, for many circumstances. Just take a clear example. All the children with neurodevelopmental problems, we're not going in there and doing brain biopsies to get the tissue, to get the RNA, to do the sequencing, to figure out their problems. So for most disorders, blood disorders, yeah, that's real easy. But for most other tissues, it is not so simple to get the right RNA. So I think it'll have its applications, but limited.
So Tuuli, if, as Bruce just sort of alluded to a little bit here, first of all, is it fair to say that probably the vast majority of diseases or pathological states are not by any means single gene diseases? There will be dozens, if not hundreds, that go into this, and how they interact and interplay with each other and with the environment may determine very much so whether you get a disease and how severely you're affected, no?
Yeah, exactly. And I think one of the important things to remember is that rare and common diseases are not kind of like two discrete classes of diseases common diseases would include things like, let's say, cardiovascular disease or asthma or Crohn's disease or these kinds of diseases. And then there are rare diseases, as we have kind of defined many times today what those things are. But this is a continuum. And also the genetic factors affecting these different diseases, they're also a continuum. There are rare variants that affect common disease risk. Common variants may kind of modify the risk of getting a rare disease as well, which is something that my lab studies quite a bit. And the sort of like being able to bring the kind of like common and complex disease research community and also clinical community together with the rare disease community, I think it's very, very important.
And one more thing, again, that sort of Bruce, I think, alluded to is, I think that given our technological capabilities now, we are being overwhelmed with a tidal wave of information. And making sense of all of this is, in some ways, a bigger challenge than anything else. How do we handle, if we start, sequencing every kid, never mind adults, how are we going to make sense of these massive amounts of information, or what we now call big data?
Yeah, so genetics and genomics has become an absolutely exponentially growing field of research, and there are a lot of different approaches that people are taking in sort of like genetic research, trying to understand these massive data sets. And there is a lot of, actually a big flow of genetic research coming from fields like mathematics and physics, and that are basically swapping to genetics to analyze these huge masses of data using approaches like machine learning and whatnot, all these fancy techniques. But at the same time, it's important to kind of keep the kind of like the biological hypotheses and the thinking about biological processes and understanding of the clinical traits and phenotypes in very close connection with the computational analysis that we are doing.
Interesting to see the convergence of all these different fields that way. Yes, ma'am.
Hi. So a question for Tuuli. So you mentioned that there's often the case that the same variant causes different disease outcomes in different patients. So my question is, what are the genetic reasons, the most common genetic reason for this situation? And also, as a researcher, how do you account for them when you're looking for the disease variant? So you want to find a disease variant. So how do you account for this situation that they are so heterogeneous across the same thing? It's different outcomes.
Yeah. Really good question. I wish that I had a perfect answer, or I wish that someone had a perfect answer, but many of the things that you asked about, we don't quite know yet. So there is the question of like what are the kind of genetic factors that contribute to this phenomenon that we call variable expressivity or variable penetrance. And so there are different kind of basically interactions between genetic variants that one can map. For example, if you have a gene lesion, a mutation that breaks the gene itself, the effect of that mutation can be affected by another mutation that affects the expression of the gene. for example. But there's a lot of work to be done in this area to find all these kind of different interactions between genetic variants. And it's one of these fields that is computationally and statistically very challenging. We need really massive data sets to be able to find these interactions. It's sort of like twice as hard as finding the initial sort of genetic lesions behind a disease. And then how we account for this in research, one of the ways to kind of get around this is, well, of course, we are kind of, all of us are aware of the phenomenon, but then also just gathering larger data sets of patients is basically one of the ways how we can account for the fact that not everyone with the variant will develop a disease, and of course, not all the people who have a particular disease have the same genetic variant, so that we can basically kind of average over larger numbers of individuals.
We're going to take one more question, but I may be opening Pandora's box here. But Jean-Laurent, let me ask you one other term that is out there a lot that sort of like, as Bruce said, if you ask 100 people, you'll get probably 200 different definitions. Epigenetics. How does that impact? See, I told you I was going to start something. How does that impact all of these things that we've been talking about? Because there's one thing, we can sequence a genome, but now there's other things that even control how that genome is expressed and what it actually does for us, or against us, or to us.
So, in a strict and broad sense of the term, epigenetics means everything that's not genetically inherited. And that's obviously a lot of processes. And we know that a number of conditions, that the expression of a number of diseases depend on epigenetic phenomena, including the impact of the environment.
These are things that may sit on or alter the genome in some way that affects the way it's expressed?
Well, it's everything that's not determined by the germline. You don't get a sunburn if you're not exposed to the sun. And, that's epigenetic.
My question is about the clinical application of genetics. So we had touched upon whole exome sequence in the clinic and gene panels that are commonly used today. But what happens and how do we teach clinicians how to use this properly? Because you can have people using these panels to diagnose or to rule out certain disorders. So if there's known risk genes, so like you can do a panel for Rett syndrome, and the patient is phenotypically Rett, but genetically, according to our panels, not. So you start treating them as having another disorder. But how do we get people to understand that it could just be that we don't know all of the genetics of that disease?
Well, that really relates to what Bruce discussed earlier and what both Tuuli and I attempted to discuss during our presentations, is the question of penetrance. Having a genotype that is being genetically affected is one thing, and having a phenotype that is having a disease, in the common sense of the term, manifestations of the disease, is another. For Mendelian disorders, there's a perfect causality and a correlation that is absolute. But for most genetic disorders, there is what we call incomplete penetrance, that is, some and sometimes most genetically affected individuals do not display the condition, the disease. Now, the mechanisms are known for incomplete penetrance for a few conditions, but for most, the mechanisms are unknown. We're sorry we cannot address your questions better. Maybe in 10 or 20 years, we'll have a better answer. Be patient.
Patience is not a virtue in scientists, I've learned, or it's not a common trait or characteristic in any case. I want to thank our panelists today for actually some really fascinating discussion. And the one thing that I think I'm going to take away from this is a quote that I will borrow. I believe it was yours, Jean-Laurent, is everything is genetic. And that's, I think, the one thing that we've learned here. If we're talking about everything from cancer to infectious diseases, if they're all involved with genetics and everything, really is
and you
genetics.
can add, and the rest is epigenetic.
And the rest is...